Hello, everyone, and welcome to this 8th episode of Cognition Conversations. Today, we'll be talking about protecting the synapse from amyloid toxicity and Alzheimer's disease. We have the great pleasure of welcoming two experts in the area of Alzheimer's research. First, Dr. Tara Spires-Jones, who's the personal chair of neurodegeneration and director of the Centre of Discovery Brain Research at the University of Edinburgh in Scotland. And then we also have Dr. Jort Faverberg, who is a neurologist and trial specialist at the Amsterdam Alzheimer's Center at the University Medical Center in Amsterdam. Finally, we're also joined by Dr. Mary Hamby, the Vice President of Research at Cognition Therapeutics. So welcome, Tara, Jort, Mary. So let's get started.
Absolutely. Hi, everyone. Thanks for having me today. I'm particularly fascinated by synapses in Alzheimer's disease because of all the things we can measure in the brain that change in Alzheimer's disease compared to healthy people. The loss of these synaptic connections in the brain tracks most closely with the cognitive decline that we see in Alzheimer's dementias. And I'm also fascinated by synapses just because they're what I've been studying for 20 years. So I'm a fundamental neuroscientist, and synapses are the connections between neurons in the brain. They're the places where the neurons talk to each other. And they're very important for learning and memory and cognition, all the things that go wrong in dementias.
Thanks, Tara. Perhaps you can tell us a little bit about how Alzheimer's is started and what the role of different amyloid proteins may be.
It's a fascinating, if horrible, disease. So we know that the brain starts changing decades before you have symptoms of Alzheimer's disease. One of the very early events in the disease is the accumulation of amyloid peptide or amyloid plaques in the brain. So this increase in amyloid, we don't really know why it occurs. In some people, you have a rare genetic mutation that increases amyloid, but in a lot of people, it's our unlucky combination of genes, lifestyle, and aging that contributes to this early increase in amyloid in the brain. And after that early increase in amyloid, one of the things we discovered in the lab is that right around every one of these big, ugly amyloid plaques that clumps up in the brain, there's a local loss of synaptic proteins, a local loss of synaptic connections. And we think that's part of the early on disruption to brain function and cognitive function. A little bit later on, after you have the amyloid accumulation, you start to see the spreading of the other pathology in the brain, which is called tau, or tau pathology, or tau tangles. And what tau is more associated with is the death of the neurons, the brain cells themselves. So one of the things we're interested in talking about today, and one of the things I've been working on, is how that amyloid protein, which is quite early on in the disease, how it actually binds to those individual synapses and causes them to be dysfunctional and eventually collapse.
Dr. Hamby, perhaps you can tell us a little bit about what we know about the mechanism of CT1812 and how we believe it is protecting synapses from amyloid toxicity.
Yes, thank you, Tony. So what we know is that TMMD7, the sigma-2 receptor, sits at the synapses, neuronal synapses, and binds to prion protein, a major constituent of the oligomer receptor, for which amyloid beta oligomers can bind. And when CT1812 binds to the sigma-2 receptor, this can lead to a displacement of amyloid beta oligomers from neuronal synapses. And what happens when amyloid beta oligomers bind to synapses is this elicits synaptotoxicity. So by removing the amyloid beta oligomers from neuronal synapses, this can prevent synaptotoxicity.
Dr. Tara Spires-Jones, tell us a little bit about your research on those interactions of amyloid, the receptor proteins, sigma-2 and CT1812.
One of the things when I came to the Alzheimer's field in the early 2000s, this is now just over 20 years ago, that we didn't know is how amyloid is damaging synapses. There had been a lot of good data to suggest that amyloid beta oligomers, so just a few molecules of that sticky amyloid peptide, that when you apply that to brain tissue, it disrupts synaptic function. And we had seen, and others had seen, that it kills synapses around those plaques. But what was still a mystery is how is it doing that? What's it binding to? And one of the things that we did in my lab when I was first in Harvard Medical School and now in Edinburgh is we developed a technique that allows us to look inside single synapses, which are incredibly small. They're so small that normal light microscopes can't see inside an individual synapse. So we had to develop a technology that was invented originally by Steve Smith and Christina Makeva at Stanford. And we adapted this technique called array tomography for looking inside individual human synapses in donated postmortem tissue. And we were able to answer this question, what is amyloid beta sticking to in synapses? Because before that time, there had been lots of ideas and lots of things that amyloid can stick to when you put it into cells in culture, for example. It can stick to lots of different things on the synapses. But we went in because of our discussions with cognition therapeutics, and we asked the question, could amyloid beta in human synapses be binding TMEM97 or the sigma-2 receptor? It was one of these many candidates, but nobody had been able to go in and look in great detail. So this study that we conducted over the past few years and just finally published this year in 2024 was using that super high resolution imaging, looking at donated human postmortem tissue, and looking at whether these little sticky amyloid beta peptides were close enough to TMEM97 or the sigma-2 receptor to be causing a signal so they could be interacting. And we found that indeed they are close enough to be generating this, what we call a FRET signal, which is a pretty cool physics way of looking at interactions of fluorophores in the brain in microscopy. So they are close enough to be interacting. And further, with collaboration with y'all, we were able to bring over some of your CT1812 compound, and we did a couple of experiments. One was on, again, dead postmortem human tissue that had been frozen fresh, and we were able to use your compound to see if we could knock A beta off of this receptor, and we could see a little bit more A beta was coming off of these sections of tissue when we had CT1812 than when we had the control. And the other thing we were able to do is treat an animal model that develops Alzheimer's disease with the compound, just like it's being used to treat patients today, and I think we'll hear a bit more about that later. And what we saw in our animal model is that we could reduce that interaction of the amyloid beta oligomers with the TMEM97 receptor. We could reduce that signal showing that they're close together in synapses in the animal model. So we're really excited about this idea of your company being able to go in there and remove something that we know is toxic to synapses from a receptor that we now know is present with oligomers in human brain.
I'm wondering if we can get Dr. Fiverberg to comment on what we observed clinically.
Yeah, thank you. So it's always great to hear like a fundamental researcher talking about what's happening in the Alzheimer brain. Of course, as a clinician, I see the patients when they have symptoms or thinking they have symptoms. So then I always imagine that indeed the synapses already lost a lot when they have symptoms. If you really want to measure what's happening actually at that moment in space and time, we use EEG. So EEG is like with sensors on your head, and then we can measure the activity. In Alzheimer's disease, you see a slowing down of the activity, and that we call the theta wave band. So that's what's going to be the dominant band in Alzheimer patients. Normally we have alpha, we were a little bit quicker waves, but in Alzheimer's disease, theta is going to take the overhand, and especially in the back of your brain. I think for the EEG, we are very experienced, but for the other measures, we're getting there, but not there yet.
We completed a study with you using EEG to measure the effects of CT1812 in people with Alzheimer's disease.
We did a clinical trial, the sequel trial, and with the mode of action of CT1812, it's obvious that you design a trial around EEG because the product is blocking the toxic amyloid from the synaptic endpoints, and the EEG measures the synaptic activity. So we powered the trial to see if we can change the EEG in a positive direction. So what we saw in this clinical trial we performed is that the brain performed better with the drug than without the drug. The slow waves that exist in Alzheimer patients was less dominant. So you saw quicker waves than slower waves. Also, the brain in different areas were more connected than patients without the drug. So in the end, we had a positive effect on the way that the brain talks to each other. So the synaptic activity was better with patients on drug than without.
So Mary, you mentioned doing proteomic analyses from our clinical trials. Can you tell us a little bit more what proteomics are, how we do it, and then also what are the different categories of biomarkers that we're looking to assess in the samples from these participants in our studies?
What we've done is we use TMT mass spectrometry, proteomics, which enables the detection of over 2,000 proteins, which we can assess the impact of CT1812 in our clinical trials when we take a cerebrospinal fluid sample from end of study compared to that patient's baseline. So from there, we can understand how the biomarker is moving across the treatment course or duration of treatment. And through those analyses in the SHINE interim analysis that was conducted, we identified that through a network analysis, Synapse was the top most represented pathway impacted by CT1812. So that was very exciting. In addition, however, we saw a robust impact on amyloid beta pathways, so amyloid beta biologies, including apolipoproteins and other clearance mechanisms, and also neuroinflammation, including an impact on clusterin. So now we have data from 2 independent cohorts with mild to moderate Alzheimer's disease, which were both given CT1812 for a 6-month duration compared to placebo, and identified that Synapse was a key biological pathway impacted. So this, we believe, CT1812 paired with the preclinical data, this is supportive of our synaptic protective mechanism.
Dr. Hamby, I'm really excited to hear those results. So synapses have 2 sides, a side that sends the signal and the side that receives the signal. And you're seeing indications that both of those are changing in biofluids. So not able to look directly in the brain because these people thankfully are still alive and participating in a trial. But you're able to detect this in the cerebrospinal fluid.
We absolutely agree. And we are also looking at plasma proteomics for our SHINE trial readout this coming summer. And that is definitely a goal of ours, is to map, the changes that we see in the cerebrospinal fluid to those in plasma, to understand if we could identify a pharmacodynamic biomarker of CT1812 that reflects a change in the brain through the plasma, measurement in the plasma.
We've completed enrollment of a trial which we call SHINE, where we're looking at changes in individuals with mild to moderate Alzheimer's disease treated with CT1812 or placebo. This trial finished enrollment in November and will be reading out mid-2024. Dr. Fiverberg is one of the investigators in this study. So, Jord, perhaps we can get you to comment on the array of outcome measures that we're looking at from this study and what we might hope to see in a trial which is positive.
I'm a clinician, so patients are central to me. So what you want is a drug that changes the symptoms they're experiencing. So in cognition, function, quality of life. And I think with the program SHINE, we measure all those things. So we measure cognition with standardized questionnaires, we measure function, and also combining that with the biology side of Alzheimer's disease. But in the end, I think this is a good phase 2 study with CT1812. And if we have a signal in the symptoms, I think this is very important. I'm very excited.
Well, this has been a wonderful discussion, and we appreciate you all taking the time to tell us a little bit about your research and how it impacts Alzheimer's disease and how it relates to CT1812.
It's a really exciting time to be an Alzheimer's researcher. We're finally in the phase where things are coming through that are helping people, and hopefully this work with cognition therapeutics will be beneficial. But in the meantime, even though there aren't very many things that are drugs that you can take right now from your own family doctor, there are still things that we can all do to improve our brain health. So we can do things like eat well and exercise and try to avoid things like head injury and smoking. And that will boost our brain's resilience, because those little synapses we've been talking about are super important, and you can actually grow more connections in your brain by leading a healthy lifestyle.
On the medical drug development side, you see a lot of things happening. Very excited about that. You see different targets, different approaches, different way we can hopefully modify the Alzheimer's disease that it's beneficial for the patient.
We have a lot of work to do ahead of us, and layering on additional mechanisms of actions as far as the therapeutic modality and approach will be really important.
It occurs to me this is really a wonderful example of all the different cross-functional experts that we need to address a problem like this. We have the basic science research coming from Tara Spires-Jones. We have the clinical aspects coming from Dr. Fiverberg. We have the industry from cognition. And all this work, or much of this work, was supported by government funding from the National Institutes of Health and the NIA, which funded the sequel trial, which Jord spoke about, and is funding the SHINE trial, which will be reading out later this year. So thank you all for joining us. We hope you learned a little bit about Alzheimer's and amyloid toxicity at the Synapse. And we look forward to our next episode of Cognition Conversations.